Hello everyone and welcome back to our channel Biodiversity and Conservation. I am Subhalakshmi Rao from Ambika Prasad Research Foundation, Odisha, India. And today we are going to study about the other two cell surface receptor, that is ion channel linked receptor and enzyme linked receptor. Because in the previous presentation we studied about GPCR, that is G protein coupled receptor, and now we will go through other two. So let's start. First one is ion channel linked receptor. These ion channel linked receptors are also known as transmitter gated ion channels or ionotropic receptors. They are involved in rapid synaptic signaling between electrically excitable cells. You can see these type of cells in neuronal transmissions. When we studied about the transmission of nerve impulse, these type of receptors are present in the nerves. So this type of signaling is mediated by a small number of neurotransmitters that transiently open or close an ion channel formed by the protein to which they bind, briefly changing the ion permeability of the plasma membrane and thereby the excitability of the postsynaptic cell. The ion channel linked receptors belong to a large family of homologous multipass transmembrane proteins. And here you can see that when the signaling molecule, like ligand, that is neurotransmitters, some neurotransmitters, when they bind to the ligand gated ion channel receptor, previously, the gate remains closed, which does not allow the passage of ions from the extracellular membrane to the intracellular membrane. But after binding of the ligand to the receptor, it opens the um, ion channels, which facilitates the passage of ions into the intracellular fluid. Thereby, it goes through the cascade of reactions, giving us cellular responses. And after the work is done, there is removal or removal of the ligand from the signaling. Sorry, the ligand from the ligand gated ion channel receptor. So after it gets separated from the receptor, the gate closes automatically. So it may be due to the um, work done after passage of the ions. It excites the cells, then the nerve impulse carried out to the other cell. So this cell will lose its activity. So the ligand gets separated from the receptor and the gate closes. Next one is enzyme linked receptors. These enzyme linked receptors are second major type of cell surface receptor after G protein coupled receptors. So these are also transmembrane proteins with their ligand binding domain on the outer surface of the plasma membrane. But instead of having a cytosolic domain that associates with the monomeric G protein, uh, there is cytosolic domain of these enzyme linked receptors either has intrinsic enzyme activity or it associates directly with an enzyme. So this is the difference between GPCR and enzyme linked receptor. And there are several classes of enzyme linked receptors, but we will mainly focus on three major classes, that is receptor tyrosine kinases, the most numerous of the enzyme linked receptors. Next one is tyrosine kinase associated receptors, and next one is receptor serine threonine kinases. So in this presentation, We will likely discuss about the receptor tyrosine kinase only. The receptor tyrosine kinases, also known as RTKs. So the ligand for RTKs may be insulin or epidermal growth factor EGF or transforming growth factor alpha TGF alpha or platelet derived growth factor or fibroblast growth factor or maybe colony stimulating factor 1. So all RTKs comprise an extracellular domain containing a ligand binding site, a single hydrophobic transmembrane alpha helix, and a cytosolic domain that includes a region with protein tyrosine kinase activity. Most RTKs are monomeric, that is fine in a single piece, and ligand binding to the extracellular domain induces formation of receptor dimers. So they are when they are in inactive form, they are found in single pieces, but after the ligand binds to the receptors, they try to form dimers so that they can carry out their activity. So here you can see after the binding of ligand, there is two tyrosine kinase receptors, so forming dimers here. And the ligand binding leads to activation of the intrinsic protein tyrosine kinase activity of the receptor, and autophosphorylation of tyrosine residues also takes place in its cytosolic domain. The activated receptor also can phosphorylate other protein substrates. There are several types of protein with which activated receptor may interact. Um, After getting activated, they can go in various paths. Some of these proteins to which it will attach, or in those pathways they can also play a major role, like the proteins are GTPase activating protein, that is GAP, phospholipase C alpha, sorry gamma, that is PLC gamma, and SRC like non-receptor protein tyrosine kinases. So there are three major pathways that can transduce a signal from the activated RTK. They are RAS MAP kinase pathway, IP3 DAG pathway, and PI3 kinase pathways. So we will discuss all these three pathways here. Like you can see here, these are the monomers of receptor tyrosine kinase. So after the ligand binds with the receptor tyrosine kinase, they try to form dimers, and after forming dimer they will start autophosphorylation and get themselves activated, and after activating themselves they go for several they go into several pathways to give us the particular cellular response. So here first one is RAS MAP kinase pathway. The ligand-induced activation of an RTK stimulates its tyrosine kinase activity, which subsequently stimulates several signal transduction pathways, including the MAP kinase pathway. RAS is a intracellular monomeric GTPase switch protein and functions in transducing signals from many different RTKs. There are three major closely related RAS proteins in humans, like H, K, and NRAS. RAS is a lipid-linked protein present in the cytoplasmic phase of the membrane, and RAS cycles between inactive GTP bound form and active GTP bound form. Um, The RAS cycling requires the assistance of two proteins, that is GEF, guanine nucleotide exchange factor, and GAP, GTPase activating protein. So RAS activations is accelerated by a protein called GEF, which binds to RAS GDP complex, um, causing the dissociation of the bound GDP. So this GEF binds with RAS GDP complex, the GDP that is attached to the tyrosine kinase, they will sorry, to the RAS, it will separate from RAS, and because GTP is present in the cells in higher concentration than GDP, so GTP will bind to the empty RAS molecules with the release of GEF. So um, this will accelerate its intrinsic GTPase activity. Binding of a ligand to a RTK leads to activation of RAS. Two cytosolic proteins, that is adapter protein and GF, provide the key links between RTK and RAS. The adapter proteins bind to an activated receptor, and then the other protein bind to them and may therefore become substrates for the receptor. The adapter has no catalytic activity on its own, but serves merely to bring other proteins to the receptor. The cytosolic receptor adaptive protein GRB2 binds to a specific uh, phosphotyrosine on an activated SRC homology domain, activated ligand bound receptors to the cytosolic GF. There is the SOS protein. So GRB2 will bind to the SOS protein, um, and SOS then promotes formation of active RAS GTP and an SRC domain, that is SH2 domain in GRB2 binds to a specific phosphotyrosine residue in the activated receptor. So the GRB2 also contains two SH3 domains which will bind and activate SOS, and SH3 binds to proline-rich motifs. The activation of a receptor tyrosine kinase may itself trigger multiple pathways. For example, activation of EGF receptor activates the RAS dependent pathway as well as RAS independent pathways, including secondary messengers. And the activation of RAS triggered by um, activated receptor tyrosine kinases are short-lived. And RAS activates a downstream serine-threonine phosphorylation cascade that includes MAP kinase, which is also called mitogen activated protein kinase. The activated RAS includes a kinase cascade that helps in activation of MAP kinase. Um, so, um, so the adaptive protein GRB2 and GEF um, and SOS link most activated receptor tyrosine kinases to RAS, and the SH2 domain in GRB2 binds to a phosphotyrosine in activated RTKs, while its two SH3 domains bind SOS, thereby bringing SOS close to the membrane-bound RAS GDP and activating its nucleotide exchange activity. Binding of SOS to the inactive RAS causes a large conformational change that permits the release of GDP and binding of GTP, forming active RAS, and the GAP gap, which accelerates GTP hydrolysis, is localized near RAS GTP by binding to activated RTKs. And the activated RAS triggers the downstream kinase cascade. The interaction of the RAF N-terminal regulatory domain with RAS GTP leads to activation of RAF kinase activity, and the hydrolysis of RAS GTP to RAS GDP releases active RAF, which phosphorylates and thereby activates MEK. MEK is MAP kinase, and the active MEK then phosphorylates and activates MAP kinase, another serine kinase. MAP kinase phosphorylates many different proteins, including the nuclear transcription factors that mediate cellular response. Then we can also see changes in the protein activity or gene expression. Here, ERK is actually MAP kinase. So it then goes through all the processes of cell in gene expression. This is also the same um, uh, sh showing the pathway of MAP kinase activity, and the activated RAS binds to the N-terminal domain of RAF. RAF is MAP kinase. So this also summarizes the same thing. Next is IP3 DAG pathway. Many RTKs can initiate the IP3 DAG pathway by activating the isoform of phospholipase C, that is the phospholipase C gamma. The SH2 domains of phospholipase gamma bind to specific phosphotyrosines of the activated receptors, thus positioning the enzyme close to the membrane-bound substrate PIP2. Um, Membrane-bound phospholipase C gamma after activation cleaves PIP2 to generate two secondary messengers, DAG and IP3. Signaling via the IP3 DAG pathway leads to an increase in cytosolic calcium to activation of protein kinase C. The next one is PI3 kinase pathway. Some activated RTKs also initiate another phosphoinositide pathway, the PI3 kinase pathway. PI3 kinase binds to phosphotyrosine residues in the cytosolic domain of activated RTKs, that is, receptor tyrosine kinases. PI3 kinase catalyzes the formation of PI3,4 bisphosphate or PI345 triphosphate, which act as the docking sites for various signal transduction proteins. Um, a primary binding target of PI345 triphosphate is protein kinase B, that is PKB, a serine kinase. Besides its kinase domain, PKB contains a pH, that is plextrin homology domain, that tightly binds the 3-phosphate in PI345 triphosphate. In an inactive form, PKB, also called AKT, is present in cytosol. In case of a rise in PI345 triphosphate, PKP binds to them and is localized at the cell surface. So PIP3 also recruit kinase PTK1, that is phosphoinositide dependent protein kinase 1, to the plasma membrane via binding of its pH domain. Active PDK1 causes phosphorylation of PKB, and the fully activated PKB then dissociates from plasma membrane and phosphorylates its many target proteins. So this is the example of receptor tyrosine kinase, that is insulin signaling pathway, where insulin receptor is a receptor tyrosine kinase. Um, it consists of two alpha subunits on the outer surface of the plasma membrane and two transmembrane beta subunits. The two alpha subunits are linked by disulfide bonds, and there is a single disulfide bond between the alpha and beta subunits also. The binding of the insulin to the alpha subunits trigger a conformational change that allows the autophosphorylation of the tyrosine residues in the carboxyl terminal domain of the beta subunits. The phosphorylated insulin receptor next phosphorylates insulin receptor substrates on the tyrosine residues. The phosphorylated IRS binds to the SH2 domains of a variety of proteins that are directly involved in mediating different effects of insulin. Um, the IS1 phosphorylated by the insulin receptor activates PI3 kinase by binding to its SH2 domain, and the PI3 kinase converts PIP2 to PIP3. The binding of PIP3 to PKB leads to the activation of PKB, and the activated PKB phosphorylates GSK3 on the serine residue, and inactivates it. So the PKB also stimulates movement of glucose transporter GLUT4 um, from the internal membrane vesicles to the plasma membrane, increasing the uptake of glucose. GSK3, inactivated by the phosphorylation, cannot convert glycogen synthase to its inactive form by phosphorylation. So the synthesis of glycogen from glucose is accelerated. Other signal transduction proteins interact with the iris, including GRP2, an adapter protein that contains SH3 domains, which in turn associate with the SOS and elicit um, activation of monomeric G protein, RAS. RAS activates the MAP kinase pathway, leading to mitogenic responses. And next example of tyrosine receptor kinase is mTOR pathway. Um, mTOR pathway is mainly involved in the cell's growth, and if the cells simply divide without growing, they would get progressively smaller. So the extracellular signals usually required for animal cells to grow and divide as well as to survive, uh, like the PI3 kinase AKT pathway that signals the cells to grow, depends on a large serine threonine protein kinase called TOR, named as target of rapamycin. In mammalian cells, it is called mTOR. So TOR exists in cells in two functionally distinct multiprotein complexes. In mammalian cells, mTOR complex 1 contains the protein raptor. This complex is sensitive to rapamycin, and it simulates cell growth by promoting protein synthesis and by inhibiting protein degradation. mTOR C1 promotes protein synthesis by phosphorylating the EIF4E binding protein 1. Uh, the phosphorylation of this binding protein prevents its binding to um, EIF4E, enabling EIF4E to promote cap dependent translation. mTOR complex 2 contains the protein Richter and is insensitive to rapamycin. It helps to activate AKT and it regulates the actin cytoskeleton via Rho family GT pages. In the presence of extracellular growth factors, activation of mTOR C1 occurs via the PI3 kinase AKT pathway. AKT activates mTOR in complex 1 indirectly by phosphorylating and thereby inhibiting a gap called TSC2, and TSC2 acts on a monomeric RAS related GTPase called REB. REB in its active form, that is REB GTP, activates mTOR, and the net result is that the AKT activates mTOR and thereby promotes cell growth. So, in summary, is with growth factor, active PI3 kinase activates AKT, which inactivates TS2, and active REB GTP activates mTOR, that is complex 1, and there we can see the cell growth. So, this is all from me today. Thank you.